There is a moment just before your hand touches a hot surface when something extraordinary happens. Something so fast it defies intuition. Before your brain has fully registered the danger, before any conscious thought has formed, your hand has already moved. Not because you decided to move it, because something inside you decided first. That something is the nervous system, and understanding it changes the way you understand what it means to be alive. Most people think of the nervous system as a kind of biological wiring, a passive network of cables that carries signals from one place to another, like electricity moving through a wall. That image is not wrong exactly, but it is dangerously incomplete. The nervous system is not a passive relay. It is an active, adaptive, extraordinarily complex architecture of decision-making that operates at every level of your body simultaneously, from the molecular to the behavioral. It is, in the truest sense, the system that makes you you. To understand how it works, it helps to start with what it is made of. The fundamental unit of the nervous system is the neuron, a cell unlike almost any other in the body. Neurons are defined by their shape, a central cell body, a long projecting fiber called the axon that transmits signals outward, and a branching web of shorter fibers called dendrites that receive signals from other cells. A single neuron can have thousands of dendrites, each one forming connections with neurons nearby or with neurons located centimeters or even meters away. The human nervous system contains roughly 86 billion of these neurons, and each of them can form between 1,000 and 10,000 connections. The total number of synaptic connections in a single human brain is estimated to exceed 100 trillion. That number is not metaphorical. It is the structural basis of memory, personality, movement, fear, language, and everything else that emerges from the 3 pounds of tissue inside the skull. But neurons are not the only cells in the nervous system. For most of the history of neuroscience, another class of cells was largely ignored. These are the glial cells, a word derived from the Greek for glue, and they outnumber neurons in many regions of the brain. For decades, scientists assumed glial cells were merely structural, holding neurons in place and offering metabolic support. Research has since overturned that assumption significantly. Evidence suggests that certain glial cells, particularly astrocytes, actively participate in regulating synaptic transmission, modulating the chemical environment around neurons, and even influencing how connections between neurons are formed and pruned. The full scope of glial function remains an active area of investigation, and there is still no complete consensus on how deeply these cells participate in cognition and information processing. What is clear is that the nervous system is not made of neurons alone. The system itself is divided into 2 major branches. The central nervous system consists of the brain and the spinal cord, the command and processing center. The peripheral nervous system consists of all the nerve fibers that extend outward from the spinal cord into every region of the body: the arms, legs, organs, skin, and face. These peripheral nerves are the channels through which the central nervous system receives information from the body and sends instructions back. Together, these 2 divisions form a closed loop of constant communication, processing information and generating responses at speeds and scales that no human technology has yet replicated. Now comes the part that most explanations rush past: the actual mechanism of how a signal moves. Because this is where the real strangeness of biology reveals itself. A neuron at rest is not electrically neutral. Its interior carries a slight negative charge relative to the outside of the cell, maintained by the selective movement of ions, primarily sodium, potassium, and chloride, across the cell membrane through specialized protein channels. This difference in electrical charge is called the resting membrane potential, and it is the stored energy that makes signaling possible. When a neuron receives enough stimulation from its dendrites, this balance is disrupted. Sodium ions rush into the cell, reversing the charge briefly before potassium ions flow out and restore it. This rapid, self-propagating reversal of charge is called an action potential, and it travels down the length of the axon at speeds ranging from about 1 meter per second in thin unmyelinated fibers to over 100 meters per second in heavily insulated ones. That insulation is called myelin, and it is one of the most important structural features in the vertebrate nervous system. Myelin is a fatty substance produced by glial cells, Schwann cells in the peripheral nervous system, and oligodendrocytes in the central nervous system that wraps around axons in segmented layers, leaving periodic gaps called nodes of Ranvier. Rather than traveling continuously along the axon, the electrical signal effectively jumps from node to node, a process called saltatory conduction. This dramatically increases conduction velocity and reduces the metabolic cost of signaling. When myelin is damaged, as occurs in diseases like multiple sclerosis, signal transmission slows, becomes erratic, or fails entirely, producing the range of neurological symptoms that characterize demyelinating conditions. The importance of myelin becomes visible precisely in its absence. When an action potential reaches the end of an axon, it arrives at a structure called the synaptic terminal, the point of communication between one neuron and the next. Here, the electrical signal cannot jump directly to the next cell. Instead, it triggers a chemical relay. The arriving electrical signal causes small membrane-bound packages called synaptic vesicles to fuse with the terminal membrane and release their contents, molecules called neurotransmitters, into the narrow synaptic cleft between the two cells. These molecules drift across the gap and bind to receptor proteins on the receiving neuron's dendrites, triggering a new electrical response in that cell. The signal has moved from electrical to chemical and back to electrical again, all in less than a millisecond. The neurotransmitters involved vary enormously, and their identities shape the nature of the signal. Glutamate is the most abundant excitatory neurotransmitter in the brain. It increases the likelihood that the receiving neuron will fire. GABA, gamma, aminobutyric acid, is the primary inhibitory neurotransmitter. It reduces that likelihood. Dopamine, serotonin, acetylcholine, norepinephrine. Each of these molecules plays a distinct modulatory role, shaping not just whether neurons fire, but how responsive they are to input, and over longer timescales, how strongly synaptic connections are maintained. Many of the most widely used psychiatric medications act by modifying the availability or activity of these neurotransmitters. The pharmacology of the synapse is, in essence, the pharmacology of mood, attention, and mental state. There is something else happening at synapses that goes beyond simple signal transmission. Synaptic connections change in strength based on experience. When two neurons fire together repeatedly, the synapse between them is strengthened. The receiving neuron becomes more responsive to signals from the sender. This is the cellular basis of learning. When connections go unused, they weaken and may eventually be eliminated. This is the cellular basis of forgetting. The phenomenon was described theoretically by the Canadian psychologist Donald Hebb in 1949, in a formulation often summarized as neurons that fire together, wire together. The underlying molecular mechanisms were worked out over subsequent decades, and the evidence for activity-dependent synaptic plasticity is now extensive. The nervous system is not a fixed structure. It is continuously being rewritten by experience. This plasticity is most dramatic early in life. In the developing brain, synapses are formed at a staggering rate. Estimates suggest that in the first years of life, the brain generates more than 1 million new synaptic connections per second. But this explosion of connectivity is followed by a process of pruning, in which excess and redundant connections are eliminated, shaping the circuits that will persist into adulthood. This pruning is not random. It is experience-driven. Connections that are used are preserved. Those that are not are removed. The environment encountered in early childhood literally sculpts the architecture of the nervous system in ways that influence cognition, behavior, and emotional regulation throughout life. The implications of this for development, education, and early intervention in neurological conditions are still being worked out. The peripheral nervous system carries its own layer of complexity. It is itself divided into the somatic nervous system, which governs voluntary movement and conveys sensory information from the body surface, and the autonomic nervous system, which regulates the internal organs and operates largely outside conscious control. The autonomic system is further divided into the sympathetic and parasympathetic branches. The sympathetic branch mobilizes the body's resources in response to stress or threat, increasing heart rate, dilating airways, redirecting blood flow toward muscles, in what is commonly called the fight or flight response. The parasympathetic branch does the opposite, promoting rest, digestion, and recovery. These two systems operate in dynamic opposition, continuously adjusting physiological parameters to match the demands of the moment. A third division, the enteric nervous system, governs the gastrointestinal tract with such independence that it is sometimes called the second brain, containing more neurons than the spinal cord and capable of coordinating complex digestive functions with minimal input from the central nervous system. One of the most persistent misconceptions about the nervous system is the idea that the brain is the only site of complex processing. The reflex arc challenges this assumption directly. When you touch a hot surface, the sensory signal travels from your fingertip to your spinal cord, where it is rerouted immediately to motor neurons that command the muscles to withdraw, all before the signal has reached the brain. The brain receives the information shortly afterward, which is when conscious pain is perceived. But the protective action has already occurred. The spinal cord is not a passive conduit. It is a site of rapid, autonomous motor control. This division of processing, some handled locally and immediately, some handled centrally and deliberately, is a recurring organizational principle throughout the nervous system. The sensory side of the system presents its own architecture of remarkable sophistication. Sensory receptors in the skin detect pressure, temperature, vibration, and pain through distinct classes of specialized cells, each tuned to a different kind of stimulus. These signals travel via different types of nerve fibers at different speeds. Sharp, immediate pain travels fast. Dull, aching pain travels slowly. The organization of sensory information in the brain follows a topographic map, with adjacent regions of the body represented by adjacent regions of cortex, a layout called the somatosensory homunculus. But the map is not proportional to body size. It is proportional to sensory importance. The hands and lips, which require fine discrimination, occupy far more cortical territory than the back or legs. The brain does not represent the body as it looks. It represents the body as it feels. Vision, the sense that dominates human experience, occupies roughly 30% of the cerebral cortex. Light entering the eye is converted by photoreceptor cells, rods and cones, into electrical signals that travel via the optic nerve to the brain's visual cortex, located at the back of the head. What happens next is not passive reception. The brain actively constructs the visual scene, filling in gaps, suppressing irrelevant detail, and assembling coherent perception from fragmented input. The blind spot, a region of the retina where no photoreceptors exist, produces no conscious gap in vision because the brain fills it in seamlessly. What we call seeing is, in a real sense, a controlled hallucination, a model of the world generated by the brain and continually updated by incoming sensory data. Memory, perhaps the most intimate function of the nervous system, involves multiple systems distributed across different brain regions. Short-term working memory depends heavily on the prefrontal cortex. The consolidation of new long-term memories requires a structure in the medial temporal lobe called the hippocampus, a fact made famous by a patient known for decades as HM, who lost the ability to form new long-term memories following surgical removal of both hippocampi in 1953. His case demonstrated that different kinds of memory depend on different neural structures, and that the feeling of continuous selfhood, of being the same person across time, is grounded in the biology of memory formation in ways that can be selectively destroyed. Studies of patients with various forms of amnesia, dementia, and focal brain lesions have continued to refine understanding of how memory is organized and how fragile that organization is. Current research is pushing into territories that would have seemed speculative a generation ago. Brain-computer interfaces are allowing signals recorded directly from motor cortex neurons to control external devices, offering potential pathways for individuals with paralysis to communicate and interact with the world. Optogenetics, a technique developed in the early 2000s, allows specific populations of neurons to be activated or silenced using light, providing unprecedented precision in studying how neural circuits produce behavior. Connectomics, the effort to map every synaptic connection in a neural circuit or brain, has produced complete wiring diagrams for small organisms and is advancing toward progressively larger systems, though a complete human connectome remains a distant goal. And the relationship between neural activity and conscious experience, the so-called hard problem of consciousness, remains as unresolved as it has ever been, a boundary at which neuroscience, philosophy, and fundamental questions about the nature of mind converge without clear answers. What emerges from all of this is not simply a description of a biological system. It is a portrait of an architecture that is simultaneously ancient and continuously new, conserved across hundreds of millions of years of evolution, yet rewritten moment by moment by the experiences of a single life. The reflex that pulled your hand from heat before you could think is the same basic circuitry that allowed your distant ancestors to survive. The synaptic changes encoding the memory of reading these words are unique to you, inscribed in a pattern of connections that has never existed before and will never exist again. The nervous system is not the passive recorder of a life already lived. It is the medium in which life is actively constructed, the place where chemistry becomes thought, where electrical patterns become sensation, where the relentless biological present becomes, somehow, experience. That transformation, from ion gradients to awareness, from molecular signals to meaning, remains the deepest unsolved question in all of science, in the fact that it happens, reliably, billions of times a second inside every living human being, is perhaps the most extraordinary fact about the body that we have yet to fully explain.